thanks to our organizers for inviting me and giving me this opportunity to present my work. I'll be talking today on the multiplex network of protein families. I've been working on this for some time with my students, particularly Pradeep and Rakhi. Pradeep is now in Mahidol University in Thailand, and Rakhi is working with me as a PhD student in Delhi University. I have presented, well, you can't hear me? Oh, I'm so sorry. Let's see if I can adjust this. Hello? Is that better? Okay. I won't, well, it's a 10 minutes talk, so I was getting ready for a 15 minutes talk, so I'll go fast forward in the first few slides because I've presented it to you many times here. Apologies to the young members of the audience who will have to catch up a little bit, and I will be happy to explain the tea time or things like that. Okay, I'll start with, of course, the basic proteins, explaining what the proteins are and what we're going to be using for proteins. Proteins are, we'll be describing protein molecules as a string of repeated units of amino acids, which are 20 in number, and the amino acids determine proteins. The sequence of amino acids are important for the 3 dimensional shapes, and the structure of protein determines function. And then the system that I've been working on is the beta-lactamase family and other protein families, but we've been studying mostly the beta-lactamase family, which is an enzyme secreted by the bacteria in response to beta-lactamase antibiotics. And this beta-lactamase enzymes destroys the amide bond of the beta-lactamase ring, which this is the penicillin. It destroys this bond and deactivates the penicillin. So it's a medical challenge to study control these beta-lactamase family and find important positions of the amino acid chain that the protein is made up of, so that it can control these beta-lactamase enzymes and hence help in the future of antibiotics. Okay, so the data, we'll be using the data to understand this or try to find the clusters which correspond to these, finding out these positions or clusters. And we will look at the data, which is in a data bank, which is this one. And it consists of 547 proteins. And you can, by certain procedures, you can eliminate the noise. And from the 547 proteins, you can sequences, you can make it into 557 and use the multiple sequence alignment to get the data matrix. Okay. So this is we do a multiple sequence alignment of the amino acids, and then of this beta-lactamase family. And then at each amino acid point, we put the property of the amino acid. And the chain is of length 248, and the number of sequences is 559. And these are the properties. This is the data matrix for the hydrophobicity property of the amino acids. This is the data for the polarity, and this is the data for the volume. Now, this is a color graph of the same data matrix. Okay, and this is the correlation matrix that will be the Pearson correlation matrix that we will be describing, okay, as a network. So we would like to characterize these the protein family in terms of multiplex networks because we have multiple interactions between the positions of the amino acids in this protein family. We can create a weighted multiplex network of evolutionary interactions between positions for the beta-lactamase family. I should just say that we layer it, that means, okay? We put each layer, we make a network and of different properties and layer it, okay? So each layer realizes a different aspect of interaction between the proteins. Okay. So this is the mathematical description of the beta-lactamase family in which we've put it in a network. Okay. The multiplex network consists of these graphs, okay, G, alpha, and E, which is layered, where alpha corresponds to layer, and it depends on the property, different properties that you have. N is the set of nodes, and E is the edges, E of alpha being different for the different layers. Okay. Okay. So the graph G alpha and E is given by the nodes N, and is given. Okay, so there are N nodes, and the multiple sequence and whatever is in the multiple sequence alignment, and the edges are described by the magnitude of the correlation elements in each for each property alpha. Okay, when C alpha ij, the correlation elements of each property, is greater than or equal to theta, which is the threshold. Okay, or it is 0. If this number is greater than theta, you put that number, or else it's 0. So that's how the network is made. Okay, so the threshold generates different networks, okay, with the same set of nodes but different edges. Because of the properties being different, these values,Cij's, are different. Okay. Now, yeah. Okay, so yeah, we'll get to this point in a minute. And this work is published now in the scientific reports. Okay, so so these are networks, okay, at different layers. So this is one layer, the hydrophobicity layer. This is the other layer. This is the polarizability layer. This is the volume layer. This is the polarity layer. Okay, so we have 4 layers. We're looking at 4 layers. You can have many layers, even more than that, corresponding to the different amino acid properties that exist in the literature. Now, you can see that for different theta values, the network changes, okay? And the network changes, and the last one gives you the connections to the strongest positions, okay? And I think I'm out of time. How much time do I have left? 8 minutes are over, okay. May I take a few minutes? Oh, 1 minute or 2 minutes? Okay, to wrap up. So these are the topological properties of each layer, and there are certain mathematical definitions of multi-links, links, okay, which are multi-links, okay, and I won't go through the definition. You can take a look at it in the paper, and which is a generalization of the link, and the multi-adjacency matrix, which is a generalization of the adjacency matrix, and the multi-degree. Okay, I just wanted to show you that these are the results, actually, the results, main results of the work. And if you look at a specific multi-degree, say 100, which corresponds to only the hydrophobicity layers, and 1001, which corresponds to the hydrophobicity and the polarity layer, which is indicated up here, you can see that if you look at theta equals 0.1, which is this threshold, you see that all the 248 positions, these are 248 positions, 233 positions are non-zero, and multi-degree K1, that is 1111. And so it indicates statistical noise. And at theta equals 0.5, only 5 nodes, okay, are with non-zero multi-degree. That is at theta equals 0.5. You can see that there are only 5 of them, okay, corresponding to the ones that, so they've all finished excepting these. So there are only 5 nodes that remain. And these nodes are very important for an experiment. You can go to the experimental literature and find that they coincide with experiments. Okay, so I just wanted to share with you this as well as some other work, and I can talk to you about this in the these other results in the tea time or lunch, whenever we are. Thank you very much for your attention.
Any questions? Okay, let us thank the speaker.